Hello, everyone. Welcome to our STEM News broadcast of things that we read about during week 4. We are STEM News Nexus at UCLA, an undergraduate organization. Subscribe to our newsletter by completing the form in the description section. Please email stemnewsnexus@ucla.edu for any accessibility-related inquiries. Now let's get started with some STEM news. So for our rapid fire section, we have 3 things. First, the first HIV vaccine trial by Moderna was held on January 27th, 2022, and it was co-administered with Moderna shots. And they said that HIV immunogens delivered by messenger RNA can induce specific classes of B-cell responses and guide their early maturation toward broadly neutralizing antibody development. Second, American placebo studies showed that vitamin D supplementation for 5 years with or without omega-3 reduced autoimmune conditions like rheumatoid arthritis by 15%. The population had 28,000 non-Hispanic whites, 18,000 Hispanics, 5,000 Blacks, 2,000 others, with a mean age of 67.1 years. And this was published on January 26th of 2022. Mice were responsive to non-hallucinogenic LSD analog as a potential treatment for depression. And this study was published on January 28th of 2022. This article on bioRxiv recently updated on January 7th, 2022, and it's titled "A Novel Metric Reveals Previously Unrecognized Distortion and Dimensionality Reduction of Single-Cell RNA Sequencing Data." Essentially, big data, or thousands of columns and rows, can be reduced by a process called dimension reduction, but at a cost of distorting the data. So usually this allows people or computers to do less calculations in less time. And if the distortion is not that big of a deal, then in downstream analysis, then people do dimension reduction. But the potential of distortion of single-cell RNA sequencing big data with dimension reduction has not been characterized, which is problematic because said distortions could create computational artifacts in cell type clustering, cell pseudo-time ordering, and other analyses. So the researchers asked, can we characterize said distortion in single-cell RNA sequencing data, and would it affect downstream analysis? So first, they utilized the metric of distortion called average Jacquard distance and tested it on stimulated data. And they realized that they could use this metric to characterize distortion. Then they used this metric on real data sets, and they revealed that data that did undergo dimension reduction did have distortion in their reduced data tables. And then they confirmed that this did affect the cell type clustering by using a different metric called the adjusted RAND index, which just basically said that the distortion in the data table did distort the cell type cluster maps. So the researchers concluded that we may need a new dimension reduction method for single-cell RNA sequencing.
Hi, everyone. This weekend, I read an article about the new methods of virus analysis and study. It is as follows. This article discovered that the ultra small size of viruses makes it challenging to visualize them using traditional fluorescence microscopy. Super resolution microscopy opens up multiple new possibilities for imaging of virus particles. And this begs the question, in what ways can we optimize the process of studying viruses, particularly viruses like COVID and HIV? Fluorescence optical microscopy, which is the topic I'm going to be discussing in this video, is the study of viruses through the use of specialized fluorescent probes, multicolor characterization, 3D imaging, and live cell compatibility. One of the principal applications of SRM imaging to virus research has been on the HIV virus, helped by the detailing understanding of the virus replication cycle.
There is now potential primordium germ cell specification agent. Essentially, once sperm and egg fuse to form an embryo that then develops into a fetus, this embryo starts to also develop its own sperm or eggs. And researchers want to look into this process because they want to understand how does a person develop fertility. And the problem is, before our recent advances in biotech, we've been limited by ethical constraints and thereby had to only experiment on frogs. Only recently were we able to experiment on monkeys. So now we are able to experiment on human cells. So to understand how the human embryo develops sperm cells, they want to first so that they can then identify potential molecular targets for treatments of infertility. Researchers asked, given that human subjects are understudied, which genes are associated with the specification of human primordial germ cells? And specification is all about this like theory of cell development, where a cell goes along a trajectory towards a cell fate. And at the point it becomes specified, it's already kind of selected its path at which fate it's going to go towards. But at the specification state, it's not entirely certain. It's kind of malleable and it can change direction and go towards another fate. So these researchers used embryonic stem cell models to find candidate transcriptionally active genes in the early primordial germ cell-like state. So essentially, they took permissible embryonic stem cells, waited for them to differentiate into mesoderm germ cells, and then induced them to differentiate into primordial germ-like stem cells. And then they did a single cell RNA sequencing at different points along the developmental trajectory and did different computational analysis and identified long tandem repeat transposons to be upregulated in more specified developmental time points along the trajectory. This long tandem repeats are basically patterns of the same sequence of nucleotides, and they are flanking this transposon sequence. And upstream, in the upstream tandem repeat, is usually a promoter. And what happens is with this particular transposon is that it gets transcribed, and the mRNA is then reverse transcribed into cDNA that then inserts into different regions of the chromatin. So they want to see how this might be involved in the specification of these germ cells. So, but first they want to kind of see, well, this is a model, so they wanted to actually test it on human cells. So they repeated the experiment on human primordial germ cells and identified the long tandem repeat transposons or retro-like transposons to be upregulated on day 14 and day 15 of real human embryos. But from then on, they have to discard them for ethical reasons. Then they decided to characterize the accessibility of the DNA using ATAC-seq, and they noticed that regions for the transcription of these LTRH5Ss becomes more open, which means that transcription factors are more able to bind. So this data is basically supporting that they are that this region is transcriptionally active in the as evidence in the scRNA data, because it's also open. Then they wanted to test the region using bisulfate sequencing to see how methylated the nitrogenous bases are, because those impede transcription factor binding sites or binding interactions. And what they notice is along these different developmental time points along the trajectory, that there is a reduction in nitrogenous base methylation, which again supports their idea that this region becomes transcriptionally active in this trajectory. Then they also noticed that specific transcription factors binded to this region more there's like an enrichment of these transcription factors in this region at this particular time point. And then they decided to then silence that region by closing it so there wouldn't be any transcriptional activity, using a long guiding RNA and a protein that would bind to it and just close it and make it unacceptable. And they essentially intercepted the differentiation as evidenced by reduced amount of these human primordial germ cells identified by fact sorting with flow cytometry. And they used all of this evidence to conclude that the long tandem repeat retroviral transposon may become active during the specification of primordial germ cells. And so there might be follow-up experiments with some sort of transplantations. However, we're still limited by doing the right thing. So yes, here are the references for this week. And I hope you have a good week.